Welcome to this lecture on signal transduction and regulation in endocrinology. Today, we're going to explore how cells communicate with each other. Imagine your body as a vast, bustling city. For this city to function, millions of cells must constantly talk to one another, coordinating everything from your heartbeat to your digestion. We will uncover the exact biochemical mechanisms and the beautiful mathematics that make this vital communication possible. To begin our journey, let us start with a simple analogy to understand how hormones work. Imagine you are standing outside a friend's house and you need them to turn on the lights. The door is locked and you cannot go inside yourself. What do? You press the doorbell. That doorbell sends an electrical signal through the walls, ringing a bell inside, which prompts your friend to act. In the human body, many hormones act exactly like you standing outside that house. They cannot cross the cell membrane, so they bind to a receptor on the surface, effectively pressing the cell's doorbell to send a message inside. Now that we have our doorbell analogy, let us look at the formal biological process. In endocrinology, this entire mechanism of pressing the doorbell and ringing the bell inside is known as signal transduction. It is the biochemical process by which an extracellular signaling molecule, like a hormone, activates a specific receptor. Once the hormone binds, it does not just do one thing. It triggers a massive chain reaction of intracellular events known as a signaling cascade. This cascade uses secondary messengers and enzymes to carry the message deep into the cell, ultimately altering the physiological state of the cell. Understanding what this process is naturally leads us to ask why it is so absolutely critical for life. Without signal transduction, our cells would be completely deaf to the commands of the body. Consider what happens when you eat a meal. Your pancreas releases insulin into your blood. However, insulin cannot enter your muscle cells directly to push glucose inside. It must bind to a receptor and rely entirely on signal transduction to tell the cell to absorb the sugar. If this signaling pathway is broken or ignored, the sugar stays in the blood, leading to severe conditions like type 2 diabetes. Having seen the biological importance of these pathways, we must now understand how they are initiated. Before any signal can be transduced, the hormone must actually bind to the receptor on the cell. We can measure how effectively a hormone binds using the principles of chemical kinetics. Interestingly, the relationship between the concentration of the hormone and the number of occupied receptors is not a simple straight line. It follows a specific nonlinear curve. This is crucial because a cell usually needs a certain percentage of its receptors activated to trigger a full biological response. To quantify this nonlinear relationship we just discussed, we use a specific mathematical formula called fractional occupancy. Conceptually, this formula tells us exactly what percentage of the cell's receptors are currently holding on to a hormone. Let us break down the symbols. Y represents the fractional occupancy, which is a proportion from 0 to 1. L stands for the concentration of the free ligand, or hormone, circulating in the blood. Finally, Kd represents the equilibrium dissociation constant, which measures how tightly the hormone binds. If the hormone concentration increases, the occupancy increases. Conversely, a lower Kd means the hormone binds very tightly to the receptor. Now that we have our formula, let us apply it to a practical clinical scenario. Suppose a patient's thyroid gland releases a hormone, resulting in a local concentration of 4 nanomolar. Laboratory tests show the receptors have a dissociation constant of 12 nanomolar. We need to find the fractional occupancy. We chose this formula because it directly relates concentration and affinity to receptor activation. We substitute 4 for the ligand concentration and 12 for the dissociation constant. Adding the denominator gives us 16. Dividing 4 by 16 yields 0.25. Physically, this means exactly 25% of the receptors are bound, which might indicate a weak signal if the cell requires a higher threshold to act. This understanding of receptor binding brings us to the next crucial phase. What happens inside the cell? Once the receptor is occupied, we see the true power of signal transduction through a process called signal amplification. Imagine a single molecule of a hormone, like epinephrine, binding to a liver cell. That one receptor does not just activate one protein, it activates multiple G proteins. Each of those produces many secondary messengers, which in turn activate even more enzymes. Because of this amplification cascade, the endocrine system only needs to release microscopic amounts of hormones to cause massive, rapid physiological changes, like a sudden burst of energy. To truly appreciate the scale of this cascade, we can represent the biological amplification as a mathematical multiplication chain. Conceptually, this formula tracks the signal as it multiplies at each step inside the cell. Let us define the variables. N total is the final number of activated enzymes. NH is the number of initial hormone molecules binding to receptors. Ng represents the number of G proteins activated by each receptor. Nm is the number of secondary messengers, like cyclic AMP, produced by each G protein. Finally, Ne is the number of target enzymes activated by each messenger. If any of these variables increases, the final biological response grows exponentially, showing how a tiny signal becomes a massive cellular action. Let us put this multiplication chain into practice with a real biological example. We are given one single molecule of epinephrine, which activates 10 G proteins. Each G protein produces 100 cyclic AMP molecules, and each of those activates 100 target enzymes. We want to find the total number of activated enzymes. We use our amplification formula because it perfectly models the step-by-step biological cascade. We substitute 1, 10, 100, and 100 into our equation. Multiplying these together yields a staggering 100,000. Physically, this means just one single hormone molecule results in 100,000 active enzymes, perfectly demonstrating why our bodies can react so rapidly to danger. Now that we have calculated the immense power of this cascade, let us visualize the entire pathway. What you're looking at is the classic G protein-coupled receptor pathway, the most common type of signal transduction in the human body. At the top, outside the cell, you can see the hormone acting as the first messenger, binding to the receptor that spans the cell membrane. Notice how this binding event triggers the G protein inside the cytoplasm. Follow the arrows to see the signal move to adenylate cyclase, which generates the secondary messenger, cyclic AMP. Finally, you see the signal fanning out to activate multiple target proteins. This diagram beautifully illustrates the physical bridge between the outside and the inside of the cell. To wrap up our lecture, let us review the key takeaways. First, signal transduction is the vital communication bridge that allows extracellular hormones to dictate massive physiological changes inside the cell. Second, this process is not random. It is strictly governed by the mathematical rules of chemical affinity and fractional occupancy. Finally, the intracellular cascade acts as a powerful amplifier, allowing microscopic amounts of hormones to sustain life. You have now covered the fundamental principles of cellular communication, which form the absolute foundation of endocrinology and medical intervention.